So, thank you all for coming to this MENDEL lecture. It's an honor to introduce our today's guest, Randy Schekman. Randy is a professor at University of California, Berkeley, and also a Howard Hughes investigator. His work has been instrumental in discovering machinery that regulates vesicle trafficking, a major transport system in our cells. Just to give you a little bit of background, he studied the enzymology of DNA replication as a graduate student with Arthur Kornberg, who got the Nobel Prize for DNA polymerase. And he received his PhD in 1975 from Stanford University. His interest in cellular membranes developed during a postdoc period with S.J. Singer at UC San Diego. In 1976, he started his lab at the University of California, Berkeley, where he has been till today for about 47, 48 years. Of course, he has received numerous awards, but just to list some of them, he has received Gairdner International Award, Albert Lasker Award, and Nobel Prize in Physiology or Medicine in 2013, which he shared with Thomas Rudolph and James Rothman. Currently, his lab investigates the biogenesis mechanism of extracellular vesicles, which are important, and probably a little bit we'll listen about this today in today's talk. And another interesting thing, so he is leading an effort supported by Sergey Brin Family Foundation to identify the major markers or the mechanisms which supports Parkinson's disease. So before I give floor to Randy, we'll present him with MENDEL medal.
Thank you. Thank you. Well, thank you for your generous remarks, Jitender. And ladies and gentlemen, I'm thrilled to be here. This is a special place and a special event. I'm grateful to my hosts for having brought me out here for the last couple of days. I've enjoyed visiting your beautiful city. I just had a tour of the Abbey. I also was able to visit the villa this afternoon and a tour of the city yesterday. And I'm looking forward to traveling to Prague tomorrow. I'm going to talk for the next hour about work in my lab for the past few years, having to do with the possibility that proteins and RNA may be trafficked between cells, not just within cells. I spent most of my career studying how molecules move around within cells, but beginning about a dozen years ago, I attracted a new graduate student who had experience with exosomes, and I found it quite intriguing because the reports then indicated that exosomes carry unique molecules, including a subset of proteins, membrane proteins, and small RNAs. And the view has been that these exosomes may be the vehicles for communication between cells, rather more complex than just hormones. Whole sets of molecules might be transmitted between cells to change metabolism, differentiation. I'm going to give you a slightly different view of how they're made, why they arise, at least in cell culture, and extrapolate to metazoans and to humans, because we have billions of these extracellular vesicles floating around in our bloodstream. So I'm going to talk in two parts about how exosomes arise, and a kind of an unusual pathway that produces them in abundance. And then I will delve into the question of whether exosomes can be functional delivery vehicles for the transmission of proteins and RNA between cells. So first, let me set the stage for what I'm going to tell you about. I want to just summarize what was known in contributions of my lab in recent years. We know that extracellular vesicles are made by a variety of cells, Even bacteria make extracellular vesicles, but much of the interest has developed in how, in metazoans, extracellular vesicles are produced and how they acquire cargo molecules fairly uniquely. Oops. There are two paths for the production of extracellular vesicles. They may bud from the plasma membrane, which is how enveloped RNA viruses do this. They are captured and pinched from the cell surface, and viruses may then spread from cell to cell, but uninfected cells produce these in abundance as well. A more complex pathway involves a branch of the endocytic process whereby down-regulated receptors may be captured by invagination into the interior of a structure called the endosome, which continues to collect a number of such vesicles, intraluminal vesicles. For the most part, these structures, called multivesicular bodies, are believed to traffic their content by fusion with the lysosome, where these proteins and RNA molecules are degraded. However, about 20 years ago, it was discovered that some fraction of these multivesicular bodies may be delivered to the plasma membrane, whereby membrane fusion, a bolus of vesicles may be discharged. And so the question is there some purpose for the delivery of these extracellular vesicles by this means? Over the course of several years, several students in my lab identified the mechanism of sorting of certain microRNAs that are captured into these intraluminal vesicles, and we identified at least 2 different RNA binding proteins that are selectively involved in the capture of certain unique microRNAs into exosomes. Exosomes themselves are somewhat uniform in structure, from 100 to 150 nanometers in diameter. They have obviously a lipid bilayer. They have certain membrane proteins that are characteristic. Often they are characterized by the presence of 4 tetra-spanning membrane proteins. One in particular that I'll describe is a protein called CD63. But they also have, in addition to small molecules and proteins, they have RNAs, fairly small RNAs, some small transcripts. The most abundant RNA species in exosomes are transfer RNAs, yRNAs, vault RNAs, but a minor chemical composition are the microRNAs, and these, as I'll show you in a moment, are fairly selective, and they differ from cell line to cell line. One form of characterization that we've used is by fractionating these extracellular vesicles. And here's an experiment that we published a few years ago from a terrific graduate student in my lab, Moraima Timosh Diaz. She used a breast cancer cell line that I'll address later on in my talk, called MB231. And she was able to fractionate 2 discrete populations of extracellular vesicles based on a difference in buoyant density. A low buoyant density population, shown here, resolved from a high buoyant density population on a linear optoprep gradient, where the sedimented particles were introduced at the bottom of the tube and then floated up over the course of a long ultracentrifugation. The low buoyant density vesicles are characterized by certain membrane proteins, one fairly unique, another CD9 shared between the 2 populations. And the high buoyant density vesicles rather uniquely contain this protein CD63. Now, in collecting and evaluating the RNA content of these 2 populations, Moraima was able to show that the low buoyant density fraction likely arises, why is this skipping? Okay, likely arises by pinching from the plasma membrane, and contains just a sampling of the RNAs at their concentrations in the cytoplasm, whereas the high buoyant density vesicles contain microRNAs that are highly selected, some with a sorting fidelity of nearly 1,000-fold. One in particular that she found is called MIR122, 1,000-fold selected. We were able to show that this sorting event could be recapitulated in a cell-free reaction, and we use that to identify the lupus law protein as responsible for the sorting of MIR122. But of course, that then begs the question about why the cell goes to such trouble to sort a minor chemical concentration of a highly sorted microRNA. Why are these vesicles made? What is their purpose? And that is the subject of my lecture today. I'm first going to tell you about the work of two current terrific graduate students, Justin Williams and Jordan No, who asked a simple question: can we measure in real time the production of exosomes in a cell culture taking samples of conditioned medium? Can we have a unique assay for exosomes that can be done quantitatively so that we can measure the physical forces that, when applied to cells, may stimulate the secretion of exosomes? So here's the assay that they devised. Using a cell line that was produced by a Japanese group, we were able to mark exosomes by using a fusion to N-luciferase at the C-terminus of CD63. Now, N-luciferase, as you know, is a really convenient enzyme to assay quantitatively. There's a luminescence assay that can be done very rapidly, and it's extremely sensitive. But in this case, they had a very clever idea, because in conditioned medium, there are pieces of membrane, there are broken cells. Very often, there are fragments of membrane that will expose the N-luciferase so that it's accessible to substrate. Normally, because of the known topology of CD63, a C-terminal fusion would expose the N-luciferase to the luminal space of an enclosed vesicle. So what they wanted to do was to eliminate the signal attributable to fragments or lysis of cells. And so they included in an incubation an impermeable membrane, a membrane impermeable inhibitor of N-luciferase, and a membrane permeable substrate, which would then only diagnose the luciferase that was present sequestered within an enclosed vesicle. So here, for instance, is a sample from a culture that had been incubated for about a day. The total signal of N-luciferase shown in the blue bar, and that fraction that is inhibited by the membrane impermeable inhibitor, shown here about 25%, and the remaining material all sequestered within the vesicles. On addition of detergent, the entire signal is eliminated. Using this assay then, and a membrane impermeable inhibitor and a membrane permeable substrate, it's possible to measure samples, really small samples, in real time over a very long period of incubation, whereas the background is very low. So bear that in mind then when I show you further characterization that shows that this is really a measure of exosomes and not merely of N-luciferase. No, wrong one. This wants to skip. Here we are. Okay. So, oh my goodness. I think I'm just going to do this manually. Yeah. So, on the left is a panel that shows that the material that's within intact vesicles remains largely in the supernatant fraction after a low-speed centrifugation, but all of it is removed from the supernatant on higher-speed sedimentations. If the material is resolved on a linear optoprep gradient, such as I showed a couple of slides ago, all of the luciferase activity sediments at a buoyancy that is exactly what was seen in a previous slide for CD63. So we believe that we're really measuring exosomes. So armed with that, they did the following series of experiments based on the hypothesis that multivesicular bodies may be mobilized to the plasma membrane by conditions that are known to stimulate the mobilization of lysosomes. It's known from the literature over many years that damage to the plasma membrane, which occurs under a variety of conditions and occurs in many tissues in our body, damage to the plasma membrane is repaired by the mobilization of the lysosome, which is drawn to the plasma membrane through the intervention of calcium-binding proteins to patch breaches in the membrane that are introduced by chemical, by toxins, or by mechanical means. They used this idea to see whether the same conditions that are known to elicit this repair response are responsible for the discharge of exosomes. So here's an experiment that shows that in 3 different forms. On the left is a very simple experiment where the calcium ionophore, ionomycin, is used to, over a very brief incubation, just a few minutes, and what they observe is a 35-fold increase in the secretion of exosomes under conditions where calcium is provided in the growth medium and the cells are rendered permeable to calcium. Really a very dramatic stimulation, not only known from the literature to stimulate the mobilization of lysosomes, but now apparently also the mobilization of multivesicular bodies. A similar response is seen when cells are exposed to a protein, a bacterial toxin, streptolysin O, that pokes holes in the plasma membrane that are large enough to allow soluble proteins to pass out of a cell, but not large enough to allow vesicles to escape. When those permeabilized cells are subsequently incubated in the presence of calcium, a quite substantial increase in the secretion of exosomes is observed as measured by this luciferase assay. And finally, just exposing the cells to mechanical shear, conditions that are milder, either with a low shear regimen by passing the cells through a narrow gauge needle, or a regimen of more stress, there is a stimulation dependent on calcium in the secretion of these exosomes as measured by luciferase. The material that is released under these conditions in the presence of ionomycin equilibrates at the same position on a buoyant density gradient, as I showed you before, that we know is characteristic of these high-density exosomal vesicles. Now, there is a literature going back over 100 years where people have explored this phenomenon of plasma membrane repair under rather extreme conditions of rupture. For example, it's known that if you simply poke a sea urchin egg with a needle, you can see a kind of explosive release of cytoplasmic content. But as these cells, permeabilized eggs cells, are incubated in the presence of calcium over time, the damage is repaired, and these eggs can even be fertilized, and they will go on to produce a normal embryo. And you can see these extrusion events from publications that were over 100 years ago. Norma Andrews and her colleagues at the University of Maryland have studied this phenomenon, and over the course of many years, they've shown that this pathway of repair results, as I indicated a moment ago, in the mobilization of lysosomes, which are drawn dependent on calcium and dependent on the presence of calcium-binding proteins to interact with snare proteins at the plasma membrane, causing these membranes to fuse and to result in at least patching. You can see this in real time by watching lysosomal membrane proteins suddenly appear on the plasma membrane by indirect immunofluorescence. It's known that these damaged membranes are then retrieved by endocytosis. They can be internalized. The damaged membranes can be repaired in the lysosome by turnover, or the lysosomes can then be recycled back to the plasma membrane. So our hypothesis was that the very same repair process that cells are known to use may employ the multivesicular body. In fact, it's known that the membranes surrounding the endosome and the multivesicular body are very similar to the membranes that surround the lysosome. So it was not too much of a leap of faith to imagine that these two populations may participate in such a repair process. Now, so we wanted to ask whether calcium-binding proteins, such as Norma Andrews had discovered to be involved in the mobilization of lysosomes, are also employed to mobilize multivesicular bodies. And so Jordan did the following experiment. He used the CD63 fusion protein, an antibody against an epitope attached to CD63. As an immunoabsorbent, the antibody was bound to beads, and a crude membrane fraction was incubated with these antibody-bound beads in the presence of or absence of calcium and addition of crude cytosol from a source rich in calcium-binding proteins. These experiments were conducted as shown in panels 1 and 2, and further, on the membrane-bound beads were treated with EGTA to elute anything that had bound dependent on calcium, and the eluted material was separated and prepared for analysis. The immunoprecipitation was very effective, either with or without calcium, because the calcium is not required for the immunoprecipitation. So you see the LAMP1 lysosomal protein being retained equally well. On inspection of the proteins that are eluted either from the control or from the incubation conducted in the presence of calcium, Jordan noticed several proteins very highly enriched and eluted by EGTA in lane 2. These bands were excised and subject to mass spec analysis. 2 different annexin proteins, annexins 2 and 6, calcium-binding proteins, already known to participate in lysosomal mobilization, were seen to be purified by this technique, and another one called calpain 3 was also recovered. That's the wrong slide. Okay. So then we wanted to ask whether these proteins that Jordan discovered are actually required for the mobilization of exosomes dependent on plasma membrane damage. So Jordan produced 2 independent shRNAs to ablate the expression of annexin 6, shown here by immunoblot. There was a very effective depletion, and copine 3 was also very efficiently depleted with their corresponding shRNAs. And then these depleted cells were compared in the presence and absence of ionomycin on addition of calcium to see if depletion of these proteins rendered the exosome secretion phenomenon greatly reduced. In untreated cells, the basal level of secretion of exosomes was not affected by depletion of annexin 6 or copine 3. However, the ionomycin incubation over, in this case, 30 minutes, showed a pronounced diminution in the secretion of exosomes for both shRNA preparations for annexin 6 and copine 3. So again, by analogy to work that had already been done with the mobilization of lysosomes, we conclude that these calcium-binding proteins are required to mobilize multivesicular bodies. Now, one last experiment that Jordan did was to use the condition of streptolysin O to produce a kind of in vitro reconstitution on addition of cytosolic proteins to restore the mobilization of exosome secretion in streptolysin O permeabilized cells. So here's the condition that he used. Cells grown in a monolayer were permeabilized with streptolysin O, and then the cells were washed extensively to strip them of soluble proteins that diffuse out through the open pores to create then cytosol depleted cells. These depleted cells were then incubated with a source of ATP, GTP, calcium, and then crude cytosol to see if exosome secretion could be restored dependent on the addition of cytosol and calcium. So here is such a reconstitution. There was, in the presence of permeabilized cells on addition of nucleotide, cytosol, and calcium, quite a marked stimulation in the secretion of exosomes by these permeabilized cells. The reaction was around 60% dependent on the addition of exogenous cytosol. The reaction was even more dependent on the addition of calcium, and control experiments with just nucleotide or permeabilized cells show a very low background. Now, in order to assess whether annexin contributed by the exogenous cytosol is required for this secretion event, Jordan took advantage of a polyclonal monospecific antibody against annexin 6. And he was able to show that the addition of this antibody reduced the signal of, this thing has a mind of its own, reduce the signal of secretion of exosomes to a level corresponding to the background signal that happens in the absence of cytosol. So this is our best biochemical evidence from a reconstituted reaction, in addition to the in vivo shRNA ablation experiment, that these annexins really are required for this reaction. Now let me summarize these observations in relation to what I told you was the known literature on the subject of mobilization. We believe that physiologic stress that may happen under normal conditions, and likely does, leads to the influx of calcium into cells. Calcium then recruits calcium-binding proteins such as annexins and copine-3 that may bind to the surface of lysosomes and multivesicular bodies, which are both mobilized and fuse at the plasma membrane, resulting in the release of exosomes. Now, let me extrapolate a bit from this. As I said, we all have billions of these exosomes floating around in our bloodstream. The notion in the field has been that these are delivery vehicles for the transfer of information. But what if they are simply a byproduct of a normal plasma membrane repair process that occurs in all organisms all the time? It's known, for instance, that when we exercise physiologists have shown that vigorous exercise results in the release of an increased population of CD63 into the bloodstream. That's almost certainly because of damage to muscle cells that occurs under conditions of vigorous exercise. There are many other tissues in the body that are subject to stress. Tumors produce exosomes in abundance. Perhaps they're also under stress to elicit this kind of a response. It may be then that the majority of exosomes produced under normal conditions are actually the result of a byproduct of a plasma membrane repair. That is I will admit, a controversial position to explain the origins of exosomes. But now I'm going to transfer to the next part of my talk, which is to offer the question of what these exosomes may be doing. Is there some way, even if they are produced by this mechanical stress, even if they're produced by mechanical stress, are they communicating information to other target cells? This is a question that was posed to us over many years, and ultimately my great colleague Jennifer Doudna suggested that perhaps we could address that question by introducing genome editing functions into exosomes that would allow us an enzymatic way and a quantitative assay for the transfer of Cas9 in a guide RNA from a donor cell to an acceptor cell. So I'm now going to tell you about the work of a postdoc in the lab, terrific Chinese postdoc by the name of Songyan Zhang. So Songyan and I cooked up a scheme that I'll show you on the next slide that has allowed us to greatly enrich Cas9 and a guide RNA into exosomes produced by a standard cell line that we use in the lab, HEC293 cells. And then I'll show you that he also has built a reporter system, in this case introduced into a breast cancer cell line, that will assess whether Cas9 contained within exosomes is quantitatively and functionally delivered to a target cell, where editing of a crippled form of N-luciferase can be read out as a means of measuring transfer. So here's the scheme that Songyan and I cooked up. It's very simple. It relies on what we know to be an interaction between streptavidin and a protein called streptavidin binding protein. In this case, streptavidin binding protein was fused to Cas9 so that the fusion protein expressed in the cytoplasm might interact with CD63 on the surface of an endosome fused to streptavidin. The two would then adhere to one another. That interaction is reversible on addition of biotin. And the thought was that if they remain bound, this would allow Cas9 and a guide RNA expressed in the cell to be carried piggyback onto the cytoplasmic surface of the endosome and then internalized by invagination into the interior to produce an intraluminal vesicle that carried an enriched population of Cas9 that might be desorbed on addition of biotin. So if this worked, when multivesicular bodies fuse at the plasma membrane, there would be a discharge of these exosomes such that the Cas9 would be sequestered within the interior of the vesicle. So we did this, and I'm going to show you in two kinds of experiments that we are pretty convinced that the Cas9 can actually be enriched and is sequestered within the interior of exosomes. Okay, so here's a typical experiment that Sung-Yan did. He grew HEC293 cells. He collected conditioned medium. He then sedimented the vesicles and particles to a high-speed pellet. This pellet was resuspended in a high concentration of sucrose and centrifuged at high speed overnight, where the vesicles are separated from particles. They're buoyant and they are collected at a sucrose interface. They can be harvested and then evaluated for their content. In 3 experiments, Cas9 was fused to a His or a streptavidin binding protein, but in a cell that was not, did not also contain CD63 streptavidin. In this case, if Cas9 were captured into vesicles, it would be just by passive diffusion into the vesicle. And you can see in the first 2 lanes that little, if any, Cas9 is detected, whereas standard membrane proteins of exosomes are detected. In contrast, when the 2 fusion proteins are expressed together so that Cas9 would adhere indirectly to CD63, using 2 independent epitopes, we see quite a substantial enrichment of Cas9, at least by immunoblot in the isolated vesicles. Now, it's also possible then, if the cells had expressed a guide RNA that bound to Cas9, to see if that guide RNA is captured in vesicles. And sure enough, in experiments where the Cas9 is expressed untethered to CD63, very little guide RNA is detected by PCR, whereas when both fusion proteins are expressed, Sung-Yan observed a 15-fold enrichment in the presence of the expressed guide RNA associated with these vesicles. Next, then, we wanted to ask if the Cas9 and the guide RNA are inside the vesicle. That actually involves a very simple experiment. In the left panel, we ask whether the Cas9 in the isolated vesicle preparation is resistant to exogenous proteinase K. In the presence of proteinase K, the Cas9 is not measurably degraded unless the vesicles are disrupted by the addition of detergent, in which case the Cas9 is degraded, as are the other proteins that are largely interior to the exosome. Likewise, we could ask if the guide RNA is sequestered inside the vesicle by exposing vesicles to ribonuclease in the absence or in the presence of detergent, and you can see that much of the RNA is protected unless the vesicles are dissolved. In other experiments, we showed that on addition of biotin to the isolated vesicles, the Cas9 is released, but remains soluble inside the isolated vesicles. It's not liberated from the vesicles, but it's retained within the vesicles. Okay, now the next part of the experiment was to design a reporter cell line with a crippled form of N-luciferase. And so what Sung-Yan did was to set up something shown here, where the crippled form of N-luciferase is only part of a cassette that starts with firefly luciferase, which can be enzymatically distinguished in its substrate specificity from N-luciferase, separated by a sequence containing a self-cleavable peptide and a Cas9 guide RNA sequence connected then to N-luciferase, but imposed in between the target sequence and the coding sequence of N-luciferase. Sung-Yan included a stop codon and a single nucleotide frame shift mutation, which would render this N-luciferase completely unexpressed unless Cas9 and the guide RNA introduced a double-strand break followed by non-homologous end joining, which tends to be error-prone, and in some fraction of the cases will produce a fusion from which the stop codon and the single nucleotide deletion have been removed to restore the reading frame, thus allowing N-luciferase to work. Now, this seems a little bit of a Rube Goldberg, but it works really well. If you expose these cells containing this fusion cassette to the guide RNA, but without any Cas9, there's very little, if any, N-luciferase expressed. On the other hand, if the guide RNA is included along with Cas9, we see a 100-fold increase in the expression of N-luciferase as measured by a ratio of N-luciferase to F-luciferase. So this is really a whopping signal, exactly the kind of quantitative signal that we would use to measure the functional delivery of Cas9 from exosomes into the cytoplasm and then into the nucleus of a target cell. So having set this all up, we were very hopeful that we would see some great signal on addition of purified exosomes to the reporter cell, but the results were a little bit disappointing. So in the first experiment, Song-Yan purified exosomes and added them in 100,000-fold excess over the reporter cells. And yet, in the presence of this, we could see these vesicles being internalized by endocytosis. They seem to remain in puncta within the cells by indirect immunofluorescence. And yet, the amount that was functionally delivered to allow genome editing of the crippled N-luciferase was pitiful, only a 50% increase in the ratio of N-luciferase to F-luciferase. In spite of that, if you then go to the trouble of expressing Cas9 and a guide RNA directly in the reporter cell, he saw a 70-fold increase in the expression of N-luciferase over F-luciferase. So it was there. It should have been accessible to Cas9, but the Cas9 was not being delivered functionally. Nonetheless, the signal was roughly proportional to the amount of vesicles that are added. So it was really a meaningful signal, but just very, very minimal. So we then went to some considerable trouble to see if there was something wrong with the way that we were isolating these vesicles. We used a sucrose buoyant density gradient. Maybe that damages the vesicles. Maybe the vesicles have to be delivered and consumed at their point of contact with the target cell. All the controlled experiments that I'll show you here gave us confidence that we weren't damaging the vesicles in some way. We took, for instance, conditioned medium from HEC293 grown cells and incubated them with the receptor cells without any fractionation, and we saw very little increase in N-luciferase expression, even after 8 days of co-culture. And finally, we did the following experiment. We cultured the donor and acceptor cells in a transwell chamber separated by a vesicle permeable membrane over the course of 6 days, where the cells could bathe each other with the vesicles that they're producing. And yet, in that time, under a variety of conditions, essentially no increase in the expression of N-luciferase was seen. So this was, of course, really troubling, but we had one more trick up our sleeves, which was to take the donor and acceptor cells and incubate them together without any physical impediment to ask whether cell-cell contact was would somehow allow the transmission of Cas9 and the guide RNA. And obviously, I'm telling you that to set the stage for the following experiment, which, as you'll see here, shows precisely this. If HEC293 cells serve as the donor and a variety of different tumor cell lines serve as the acceptor, we saw in all cases quite a substantial intercellular transmission of Cas9 and guide RNA. Noteworthy, the Cas9 and guide RNA didn't have to be tethered to CD63 at all. Likely, it seemed to be able to be transmitted between these cells even just by diffusion. Now, in contrast to the behavior of these pairs, if Song-Yan constructed HEC293 cells to serve both as a donor and an acceptor, no intercellular transmission was observed. That is, there's some asymmetry in the contribution of these cells, where HEC293 is a terrific donor but a terrible acceptor, whereas the tumor cells are good acceptors. We found that the tumor cells are also good donors. So there's something different about these tumor cell lines compared to this otherwise transformed but non-tumor-forming cell line, HEC293. Well, we asked, could it be that there's some intimate contact where the cells form something equivalent to an immunologic synapse through which vesicle communication might be directed very efficiently to allow more efficient uptake? Such an interaction would require endocytosis of the vesicles that are produced by the bound neighboring cell. So we asked, is this intercellular transfer that we observed dependent upon endocytosis? And using inhibitors and knockdowns, we saw no effect on inhibition of endocytosis in this transfer. So we don't think it's communication mediated by vesicles at all. Instead, we think that this may be mediated by tubular connections that are known to form between cells in cell culture. This has been reported over many years in the literature. There are such connections that form that in many different cell lines. They're very often thin tubular connections called tunneling nanotubes. And in many examples in published work, people have seen the movement of messenger RNA, of proteins, of protein aggregates, even of organelles such as mitochondria across this junction from one cell to another. Now, what literature is known on this can be summarized here. It's known sometimes that these tubules emanate from both donor and acceptor cells. Sometimes they come from one cell and they touch and adhere but do not fuse with the target cell. An example of this are cytonemes that Tom Kornberg has characterized, and he has suggested maybe delivery vehicles for the transfer of ligands to stimulate target cells by delivering them directly to the cell surface but not requiring membrane fusion. On the other hand, there's also evidence that sometimes these intercellular tubular connections bind and then resolve by a membrane fusion reaction, which produces an intercellular continuity, allowing cytoplasmic flow. It's known under these circumstances of open-ended connections that actin filaments project across the tubular junction, and it's believed that these actin filaments may be responsible for the transmission of either soluble proteins or organelles that move perhaps back and forth between cells, at least in cell culture, and likely in tissues, although it's more difficult to establish this in tissues. So now in the remaining time, I'm going to tell you about our effort to visualize such tubular connections in the experiment that Song-Yan has conducted. And he's marked both the donor and acceptor cells, as you'll see in the next experiment, distinctly. The donor cells, HEC293, are marked by the presence of Cas9 GFP, so there's an overwhelming signal of GFP in the donor cells. And the acceptor cells are marked by the addition of a fluorescently tagged form of wheat germ agglutinin that's added to the co-culture. And over time, Song-Yan has observed tubular connections, occasionally resulting in fusion and with the transmission of material between the cells. One particular example of the transmission from the donor cell can be seen in the acceptor cell here, where the donor cells were labeled with an M cherry fused to CD63, coming only from the donor cell. And even though cytoplasm isn't rapidly moving from one cell to the next, this organelle is seen projecting quite considerably into the cytoplasm of an acceptor cell. We can watch these junctions in real time, and if this movie works, you can see this. So here is a cell that is about to form a junction. Imagine you're in the Sistine Chapel and you're seeing God touching the figure on the ceiling. So here we have an arm extended from the donor cell, and in this case, it's apparently an open-ended connection. These connections come and go. Sometimes they're quite stable. Sometimes they're ephemeral. They break very rapidly after forming. And sometimes the connections form and they don't resolve to fusion, and they stay remaining sometimes for hours. Here is one such connection now visualized at higher resolution by correlative light and electron microscopy. So Song-Yan captured this particular cell where the open-ended fusion can be seen. At the level of fluorescence, this sample was marked and then processed for thin section electron microscopy, and you can see the continuity here. It takes a great deal of patience in this case to be able to cut sections that cut both through both donor cells and through the thin tubular connection, but he's a very patient person, and he saw this image. He marked the contours of the plasma membranes that are contributing to this fusion event and found a discontinuity, likely where fusion occurred, allowing flow of organelles one way or both ways. Now, I told you that HEC283, though it's a good donor, is a terrible acceptor. So what's going on? Well, we've seen, and indeed the literature contains published images of HEC293 cells with tunneling nanotubes apparently connecting adjacent cells. And yet there was no transmission. So we asked, is there something wrong? Is there not any evidence of open-ended membrane fusions that would be required for the transport of material between adjacent HEC293 cells? And so here's an experiment that Song-Yan conducted with HEC293 cells serving both as the donor and as the acceptor. And over a very long period of time, several hours, patiently he observed a logjam at the point of contact between the donor and acceptor. You can see this in higher mag. And even after a couple of hours, there's really very little evidence that these contacts between adjacent HEC293 cells can resolve by some membrane fusion event. So we think this is the explanation for the failure of these cells, and it suggests that the tumor cells that serve as effective acceptors may have some additional machinery that's necessary to create this open-ended continuity. Now, what could that machinery be? We think that the failure of exosomes to deliver content, but the success of this fusion event in using a tumor cell line must be attributable to a role for a membrane fusogen. We suspect that exosomes are not terribly efficient because they generally lack any of the known membrane fusogens that are present in our human genome. There are several membrane fusogens. One is involved in sperm-egg fertilization. Another pair of proteins implicated in fusion are responsible for the fusion of myoblasts to create differentiated muscle cells. And finally, a very interesting fusogen called syncytin, which is encoded by a retrovirus that was integrated tens of millions of years ago into the human genome. This virus-like fusogen is expressed fairly uniquely in the early embryo, and it's required when a blastocyst forms a layer of cells on the blastocyst surface called trophoblasts. These turn on the expression of this cell surface fusogen called syncytin to create a syncytium that gives rise to the placenta. Now, although that expression is rather limited to the early embryo, it turns out that tumors upregulate the expression of syncytins. And this may explain some behaviors of tumors in being able to harvest material from adjoining cells. So actually, let me go back just to the HEC293 to convince you. We can also, Song-Yan also observed cells that form these tubular connections, but they appear not to be functional. Indeed, when he looked at higher resolution, he could see that the junction between adjoining HEC293 cells did not result in a fusion event. The cytoplasms remained discontinuous. So we turned our attention to this molecule syncytin because it turns out it's expressed in the breast cancer cell line that we've investigated. Indeed, it's generally found in tumors and in a variety of transformed cell lines. So here is, I think my slides have a mind of their own. Here is a diagram of what syncidin looks like and how it may engage cells in cell fusion. Syncidin is a type 1 membrane protein. It's characteristic of a family of viral fusogens. It has a C-terminal cytoplasmic domain, a single membrane anchor domain, a site for palmitoylation near the membrane anchor domain, a furin cleavage site that's acted upon when the protein traverses the Golgi apparatus. This furin molecule cleaves at this position to create a soluble, secreted N-terminal domain that is responsible for binding syncidin to a receptor protein present on the surface of an opposing cell. It's believed that the receptor binding domain remains covalently attached, possibly by a disulfide bond, to the active fusogen portion of syncidin. The receptor binding domain interacts with one of two different receptors on an opposing cell. Once they engage, the receptor binding domain is believed to dissociate, releasing the active, now mature N-terminal fusion domain, which inserts into the adjoining bilayer to catalyze membrane fusion. This is very much like many of the viral fusogens, but this is endogenous. This is really the only viral-like fusogen that's endogenous to the mammalian genome. There are two paralogs of human syncidin. They are corresponding, those sequence different fusogens in the mouse. They are called syncidin A and B. And so what Songyan did was to ask whether by knocking down syncidin 1 or 2, if he could eliminate the intercellular transfer of Cas9. And so in this experiment, you see that a partial effect on reduction of syncidin 1 is observed, but a quite substantial reduction in the efficiency of transfer is observed when syncidin 2 is knocked down. And in both cases, particularly with syncidin 2 knockdown, that knockdown can be compensated by the expression of mouse syncidin A in the human syncidin 2 knockdown. So this result, the substantial reduction in efficiency of transfer and its repair by expression of mouse syncidin, gave us some confidence that syncidin in the acceptor cell may be the responsible fusogen. So finally, then, it was possible to observe in cultured pairs of HEC23 cells and MB231 cells from which syncidin 2 had been knocked down, that these cells fail to form functional junctions, just like in the cultures of HEC23, where it's both donor and acceptor. We found when this syncidin is knocked down in the acceptor cell, very little, if any, evidence of fusion can occur. This was followed, of course, by correlative light and electron microscopy, where Songyan was able to capture one such image where the acceptor cell had syncidin knocked down. And we could see quite broad contact between the donor and acceptor cell, but no apparent breach in the membrane. The cytoplasms are of different density, and thus there is no effective transfer of cytoplasmic material. Okay, well, let me summarize this and some other observations and conclude what I've shown you today. We've shown that this intercellular transfer mediated by tubular connections requires syncidin, likely syncidin 2. In other experiments, we've shown that a receptor that is known to interact with syncidin 2 is required. And once the fusion occurs, we believe that either endosomes or soluble dissociated Cas9 is free to diffuse or possibly to be trafficked along actin filaments into an acceptor cell. The Cas9 has been provisioned with a nuclear localization signal so that it can be imported to act on a guide RNA, on a target sequence, allowing the production of genome-edited and functional N-luciferase. Well, there are many interesting implications of what I've said. particularly with the possible role of syncidin in tumors. It's known, for instance, from a very small literature that's available now, that if you look at a xenograft transplant of a tumor into a non-transformed animal, you can observe at the margin tubules that emanate from the tumor into the margin, where occasionally a tubule will connect to a cell, a non-transformed cell in the margin. And one report has shown that mitochondria appear to be hijacked from the adjoining cells and delivered back into the tumor cell. So it's quite interesting that the syncidin expression that's upregulated in tumors may serve a purpose in allowing tumors to survive, to be nourished, to remove contents from other cells. Another interesting implication of what I've shown is that there is some evidence that tumors, certainly they produce a lot of exosomes. There's some evidence that exosomes arising from tumors migrate within the body and may then be taken up by tissues to create what is referred to as a pre-metastatic niche, into which cells from the primary tumor may migrate to allow metastatic cancer to progress. And if that's actually true, it may be mediated by syncidin that's expressed in the tumor cell line. Perhaps syncidin can be functionally taken up by exosomes and then will allow these exosomes to deliver content. This is something that we're looking at now very actively in the lab. Well, let me leave you with the people in the group whose work I've described. We started with the work of a former graduate student, Moraima Tamosh-Diaz, who was successful in resolving different populations of exosomes. I told you about the work of these two terrific graduate students, Justin Williams and Jordan No, who showed that exosomes are mobilized likely as a byproduct of plasma membrane repair. And finally, the work of this terrific postdoc, Songyan Zhang, who's done all of the work on these, the functional analysis of exosomes and the role of intracellular tubular connections, possibly mediating intracellular traffic. Now, if you will indulge me for a minute, another favorite topic of mine is to talk about the problems that we all face in publishing our most prominent papers in the biomedical literature. I've served over many years as a journal editor, and I've developed quite a strong attitude about the toxic influence of commercial journals that we now have to pay large sums of money to have our work published in an open access format. Well, I'm here to tell you that these organizations are pirates, and I'm not the only person who said this. Elsevier is the largest publisher of scientific literature in the world. They have the highest profit margin of any company in the world, not in terms of absolute dollars, but in profit margin. After all, they rely on our free labor to do the work, to evaluate the papers. They pay their professional editors next to nothing, and they charge us an arm and a leg to have our papers published in an open access format. This has, I think, for many reasons, had a very detrimental effect on the literature, and I think the matter is only getting worse, and I'm not the only one. Apparently, one particular group of editors of a whole journal resigned en masse in protest over the extortionist levels of charge imposed by Elsevier on its customers. Well, I know, because I talk about this wherever I go, and I know that many of you will say, you'll roll your eyes and you'll say, but I'm a victim of the system. I must publish in these journals if I'm going to have a successful career. So let me advertise for you a new journal that is about to hit the press that I know many of you who are biochemists and are interested in reductionist scientists will find greatly appealing. And it's published by the Nature Group. It's a new journal called Denature. And you can read in the pages of Denature even more about CRISPR, and you can pretend that you understand why it's such a big deal. Yet another crystal structure, but don't worry, cryo-M will soon replace them, some more bullshit science. And the reason why these people got published in Denature is they had better equipment. Thank you for your attention.
So thank you for the great talk. Questions?
Thank you. It was a really impressive talk. So you nicely described these two ways of intercellular communication, through exosomes and through these nanotubes, right? So I am always puzzled, like, so how does the cell decide which way it will communicate?
Yeah.
Whether it will form exosomes or
Yeah.
whether it will form nanotubes?
Yeah. Well, my view, which is a minority view in the field, is that most exosomes or extracellular vesicles are not involved in communication at all. From what I said in the first part of my talk, I'm guessing that they are simply a byproduct of a normal plasma membrane repair process. After all, multivesicular bodies were known for years to simply deliver their content by fusion to the lysosome, where the vesicles will simply be degraded to amino acids and sugars and nucleotides, and those reused by the cell. Some fraction of them happen to be diverted to the plasma membrane. Maybe it's because plasma membranes are normally very often under stress, and they must be mobilized to serve as an added benefit to the lysosome to repair plasma membrane. If that's true, and there's some physiologic evidence to support this, then the exosomes are not being delivered to the cell. They're not being secreted for any particular purpose. And so when you try to see if they're functional, efficient delivery vehicles, they aren't. There are some examples where we can see some functional delivery, but I'm guessing it's very inefficient. And I think the problem is that in order for an exosome to be a functional delivery vehicle, there has to be some catalyst of membrane fusion. And most cells don't express a membrane, an exoplasmic membrane fusion protein. So most cells aren't going to be capable of producing an exosome that can deliver anything. So this is not a popular view, but at this point in my career, too bad.
Okay, thank you.
Anyone else?
Did you try to fuse the syncytin to the GP63, similarly like you do with the...
Try to fuse it to...
fuse the GP62 to force it into the exosomes, whether you would...
Yeah, that's a great approach. We're doing those sorts of things now. Trophoblasts, there's a cell line, trophoblast cell line, which you can show massively, the cells will massively fuse when you stimulate it with an agonist that promotes this fusion reaction. And we're trying to see if exosomes produced by trophoblasts are capable of delivering content. And so, but those experiments are just underway. It may be that if we directly fuse it to CD63, we'd see a different effect, but we haven't done that yet. There's a question in the back there.
Thank you for the talk. Let me go to the beginning where you mentioned that you described that for 2 microRNAs you have specific mechanisms for them to get enriched in exosomes.
Yeah.
And you shared your skepticism about the signaling function. But could it be that maybe some cancer cells use exosome microRNA content to actually get rid of the endogenous microRNA?
Yeah, you're right. It could be that it's just that these microRNAs are it's a disposal pathway just to reduce the cytoplasmic concentration of undesired microRNAs.
The capacity of the system is big enough to do that?
I don't know. But we do know that their microRNAs are only a tiny chemical fraction of the RNA content of exosomes. So clearly the exosomes have the capacity to package more RNA. Whether they have the capacity to package more microRNAs, I don't know. But you gave me an example earlier today of B cells that have an abundant microRNA that seems at least a good fraction of it to be secreted, so maybe they do.
Thank you. It was a very nice talk. I have just one question. It's more like the orientation, perhaps, in and out. I mean, if I understand you right, the exosome would be somehow logical to fuse with a cell, and the content gets into the cytoplasm. While when you have the tunnel, you don't change the orientation.
No, it's the same orientation. It's the same topology. An exosome has, as its topology, the extracellular surface is equivalent to the extracellular surface of, of a plasma membrane. It's the same topology. A secretory vesicle inside the cell has the opposite topology. So when secretory vesicles fuse, it's the cytoplasmic surface of a secretory vesicle fusing with the cytoplasmic surface of the plasma membrane. That is what is catalyzed by snares and other things. Those are abundant and well represented throughout the cell. The other kind of fusion reaction occurs on the other side of the membrane, and there are very few examples. And generally, most cells don't have such membrane fusogens. Viruses have evolved their own fusogens that are essential for the delivery of viral RNA.
In your case, you have the CD63, right? You have an orientation of the Cas9.
Yes.
You have it on the vesicle that doesn't enter the donor cell unless it makes the connection, like in the last model that you showed. And it's still the whole vesicle transported through the tunnel. That was kind of for me. So how
Let's go to that slide.
Perhaps I missed it, I don't know. Yeah, exactly, there under.
Yeah.
There the orientation is not.
So we propose that the Cas9 that gets through these intracellular junctions is either freely soluble in the donor cell, or it's tethered on the cytoplasmic surface of an endosome. This is an endosome, not an exosome. The exosomes are inside the endosome. So the orientation of an endosome is the cytoplasmic surface of the endosome is the exterior, and that's the site to which we had tethered Cas9 to the C-terminus of CD63. The C-terminus of CD63 is inside an exosome, but it's on the outside of an endosome. This is a subject that I pound on my sophomore students when I teach introductory biology. I know it's very confusing.
Okay.
Can you comment a bit on the last slide, where you see the future of scientific publishing?
Teenager, yeah. Well, yeah. I'm troubled. These commercial journals have become even more powerful, and I'm very concerned about the way they curate the scientific literature. They hire professional editors. These are people who were trained as scientists, but they now are called in to serve the business plan of the commercial publisher. And they are called on to review, to handle papers well outside of their own expertise. And so they are then asked to judge the comments of referees who are our peers. Often the comments are disparate, the referees have very different opinions, and these professional editors have to somehow judge the disparate comments of active scientists. And I just frankly don't believe they have the judgment to be able to do this. So if many of you will have experienced, when you submit a paper to one of these journals, you'll get, sometimes a half a dozen reviews with a bunch of different comments that don't agree with one another, and you're called on to reconcile this to get the paper published. I think the system that's used by these commercial journals is really flawed, and we see in the end that there's a lot of garbage that gets published. And frankly, the problem, as I see it, is that the professional editors aren't able or willing to put their name on the paper when the paper gets published. When you publish in a journal that's run by a scientific society, the editor, the member of the editorial board, who's handled your paper very often will have his or her name on the paper, and that's a stamp of approval by a person who's a peer. Whereas in the commercial journals, Cell, Nature, and Science, it's not a commercial journal, but uses the same mechanism, you will not see the name of the person who handled your paper. They are unwilling or unable to provide that stamp of approval. I think that's a very serious flaw in the way they manage the literature. And we are all victims of this system. And I wish there were a way. I tried with eLife to build up a journal, but then I decided to do something else after a few years, and my successor has made a mess of things, so I'm not happy about that. But you know, when we, collectively, as a group of scholars, refuse to allow these professional editors and these commercial journals to run our lives, then we'll make things better. But you know, I can say that, but others don't feel comfortable doing that. I wish I had a solution for you, other than just don't publish in these journals. In the back. Yeah. 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 Well, right. I think the institutions are in a conflict of interest in pushing their own scholars and publishing the papers of their scholars, so it should be independent groups. I think scientific societies are the perfect vehicle for the publication of the literature, and the policy in my group is we will only publish in journals where all of the decisions are made by active scholars. And these are published by, you know, societies. There are a variety of societies. Most journals are run by scientific societies or not-for-profit organizations like PLOS. These are journals that I trust. They're not perfect, but they're not publishing because of a business plan. They're publishing because they're serving the scholarly community. Yes? Yeah. 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 Well, open access is the solution for reuse of data, and many of us feel very strongly that when papers are published in open access, they should be with the most generous license. The CC-BY open access license is the most generous for reuse of what's published, and all that's required is that the original paper be cited. There's no transfer of cash or anything. So journals should not own the literature. It should be owned by the scholar who produced them. And so I'm a big supporter of posting things on the bioarchive. I think everybody in biomedicine should be posting a paper as soon as it's ready to be submitted for publication, and that is understood that it's not peer-reviewed, but it's out there for people to look at and comment on. There's been many decades of success in the physics community with the physics archive, where it's expected that people will post their papers on the physics archive, and then we'll eventually pull it off and publish it in a regular journal where it's peer-reviewed. I think that's a perfectly good way of doing it. And even most of the commercial journals now accept that you're going to post your work on an archive to begin with. I think that's a really good trend. But the commercial journals fought aggressively against open access because it was a threat to their profit margin. Now that they've realized that they continue to make too much money, even with open access, we're now all victims having to pay large sums of money to be published in these commercial journals. It costs the equivalent of $11,600 to publish a paper in Nature, not just in the main journal Nature, but in most of the titles that have Nature in the title. That's a complete ripoff. It doesn't cost anything near that to actually process a paper for publication.
Okay, more questions? Okay, then let's thank Randy for the talk.